Hello everyone and welcome to the third installment of your recorded lectures for Biology 303 this fall 2013. Excuse me. I will post it when you see this uh, recording on the Blackboard site. You'll see these associated slides that will be titled Human Microbe Interactions and in parentheses Normal Flora. So what we'll be talking about today is describing the normal flora of the human body, the microorganisms associated with different compartments of the human body, as well as we'll sort of discuss the role of some of those normal flora components in causing some diseases. And then we'll have a second separate section on patterns of pathogenicity that are common among some of the pathogens we've talked about already in terms of STDs and other organisms. And then we'll talk about some specific mechanisms of virulence that are unique to certain classes or kinds of microbial pathogens, and specifically there we'll talk about bacterial toxins. So again, this recording and this set of slides will deal only with the normal flora, and there'll be a second and separate recording associated with bacterial pathogenesis and toxin production. I'll just quick check here, make sure we're recording okay. Looks like we are, and we will move on to this first set of slides. Oops. Excuse me. This section is essentially describing beneficial interactions between microbes and humans. So these are not disease-causing organisms per se, although some of them can lead to that. Specifically, we'll talk about components of the human normal microbial flora, and we'll look at the distribution of those organisms across different compartments of the body as described or outlined here. So as we've talked about in the context of just understanding basic microbiology over the course of the semester, most microorganisms are benign and do not cause disease in humans. And many of these organisms that are associated with humans are not disease-causing organisms, can actually play a role in preventing disease, and these are commonly described as the normal flora of the human body. And that flora differs across different compartments and tissues of the human body. So how do these associations occur, or are they created? We are colonized at birth and soon thereafter by microorganisms via a couple of different routes of exposure. One of those is birth canal, so the surface of the human body picks up a variety of microorganisms. If you have a baby that's born sort of via natural childbirth rather than cesarean section, you are exposed to the normal flora of the mother during passage through the birth canal, sort of an initial inoculation of the skin surface with a variety of organisms that are found in that portion of the mother. We are also continually inoculating our gastrointestinal tract, although when we are born, our gastrointestinal tract is sterile, and the initial colonization of the gastrointestinal tract, at least for some of the organisms that are identified there, have been linked to breastfeeding and also eating of solid food, although in the first few months and up to a year or so, depending on individual behavior, the primary source of food resources comes from breast milk. And breast milk contains numerous beneficial microorganisms, and this is relatively new understanding of the distribution and sources of inoculation of the human gastrointestinal tract, and that's that it used to be thought that breast milk was sterile, and actually it contains quite a few different species of classes of both known and well-described obligate anaerobic organisms, and that includes the genus Bifidobacterium, and then fermentative gram-positive rods known as and the genus Lactobacillus, amongst other things. And then you see a citation there from a recent study from 2008 describing the presence and distribution of different Bifidobacterium species and subspecies inside milk and how they become part of the human infant microbiome. Many of these organisms can play a role in assisting in digestion as well as development of the gastrointestinal tract for harvesting resources from food that is ingested by the human and translating that to resources or nutrients that are available in picked up by the circulatory system. There also are some connections between the microflora of the human gastrointestinal tract that seem to play a role in limiting or preventing potentially autoimmune-based diseases like type 1 diabetes. And there's even a recent study, as recent as last week, although I won't show you the slide here or a title slide here. There's a recent publication in Nature that describes in mice, not in humans, but the prevalence of different microorganisms in the gastrointestinal tract as being connected to the onset of autism-like symptoms in mice. So clearly there is a connection between these members of our gastrointestinal community that act as mutualistic components of a healthy individual human, and those are acquired in a couple of different ways: initial colonization through ingestion and surface colonization during birth, ingestion of milk, and colonization during birth, and then continual modification of that gastrointestinal tract community through diet and other sources of inoculation. Here is a list of some of the different compartments of the body that we'll talk about and the corresponding lists of major genera that are found on different compartments of the human body, as discussed and presented in our laboratory presentations last week. You may have noticed that some of these genera associated with the human skin surface, like Micrococcus and Staphylococcus, are more or less common, or I should say commonly isolated under the conditions which we used in the laboratory setting. I would you see that also so in the mouth there are some genera there that a number of folks in our class were able to isolate from the upper respiratory tract and cultivate and then describe in the laboratory. And we'll talk about, and we'll see also in the lower respiratory tract, there may be some members of the normal flora that are sort of extensions of the upper respiratory tract, whereas other things might be more representative of, say, disease states, although there are numerous bacterial species in the upper respiratory tract and the lower respiratory tract, though that tends to be sterile. We see a much longer list of typical genera present in the gastrointestinal tract than the urogenital tract, but another thing to note here is that although there are some things that are common between the upper respiratory tract and upper gastrointestinal tract, like Staphylococcus, many of the organisms listed here are unique to the gastrointestinal tract, and so much so that you may notice that some of these genera, like Bacteroides, Clostridium, Enterococcus, Methanobrevibacter, many of those are obligate anaerobic organisms that thrive in the anaerobic environment of the lower gastrointestinal tract, but would not necessarily be able to make a living on the skin surface or respiratory tracts. So how are these interactions occur? Similar to some of the processes we've discussed earlier in the semester. We've talked about typical biochemical pathways that allow different organism classes to access carbon resources that are typical of, say, mucus that is produced by mucous membranes or epithelial cell lining in the human body or other animal systems. Animals can provide a favorable environment for growth and enrichment, cultivation of certain organisms, if they are able to access those unique carbon and nitrogen resources that are either produced by or held within the animal cell systems. Typically, this process occurs similar to the kinds of processes we talked about for viral replication. There's an initial contact between microbial cells and an epithelial cell lining or other cell surface. Those microbial cells are able to attach and grow, particularly they may be able to feed off of components of the mucous lining, or they may receive other resources that are directly exuded by the epithelial cells to provide a substrate for growth. And then sometimes either associated with disease or the normal mutualistic development of relationships between members of the microflora, there may be movement from the initial binding sites through the gap junctions between epithelial cells and either into the circulatory system or close thereby, or maybe even into the host cells, or just residing between cells in those gap junctions. That can be either initial stages of a disease state, or there are also members of the human microflora that move between those gap junctions as part of establishing their normal mutualistic relationship with the human host. So let's look at some of these different compartments. We'll start with the skin. Skin is typically dry and acidic and does not support the growth of a wide array of microorganisms. However, at certain locations on the skin surface where there are sweat glands, you can see in the image there, those can be sources of low molecular weight organic acids that are released from those sweat glands and either are associated with hair follicles or are exuded out on the skin surface. And those low molecular weight organic acids and oils can be food substrates for heterotrophic growth by a suite of organisms that can tolerate the dry and acidic environment of the skin surface. And so there are key environmental characteristics that select for different members or drive the selection for different members of the skin microflora, and those include temperature, humidity, pH. And there are host factors also that influence the growth conditions on the skin surface, and some of those are related to things like personal hygiene, diet, and age. That can vary the production of the type of oils and amount of oils from those sweat glands, and therefore sort of selecting for different classes or groups of microorganisms that might inhabit the skin surface. We look inside the oral cavity, we see a different type of surface on which organisms are able to or need to be able to adhere to and then take advantage of. This is a much moister environment, right? A place where there's typically a constant supply of organic matter due to just mastication or chewing of food by the human host. Inside the saliva that's produced in the oral cavity, there are oftentimes enzymes that play a role in the initial decomposition steps of breaking down food resources, but also those enzymes can play a role in acting as antimicrobial agents that limit the ability of microorganisms to adhere to the enamel surfaces of the teeth or the skin surfaces of the gums, and those enzymes play a role in limiting biofilm formation, for example. The tooth surface consists of a mineral matrix that surrounds sort of a living dentin or bone surface. That hardened enamel surface plays a role in enabling us to digest or masticate food, but also is a relatively smooth surface initially as it's produced that limits adhesion by microorganisms or can not prevent it 100%, but limits the ability of some organisms to adhere to that surface. However, some organisms are able to stick and colonize those enamel surfaces if they are able to grow to high enough densities to where they form thin biofilms. The base of those biofilms can become anaerobic just due to limitations in oxygen diffusion or recharge of oxygen through that biofilm surface. And if those organisms are provided with enough heterotrophic growth resources, they can stimulate growth and not respiratory activity, but fermentative activity that can result in acid production and higher levels or concentrations of acid being collected on the enamel surface and a lowering of the pH. This can result in a drop in pH and essentially a dissolving of the enamel surface. That is the initial stages of a cavity formation or dental caries formation. And so these next set of bullets sort of describe that process, right? So this initial colonization, growth of a biofilm or plaque, creating anaerobic sites, and this allows the formation or the promotion of fermentative activity and the production of organic acids. So there is a class of, or a group, a subset of the human microflora, oral microflora, that oftentimes play a role in this process. This includes Streptococcus sabrinus and Streptococcus mutans that can live fermentatively, right? They are lactic acid fermentative bacteria. Feed them enough sugar and under anaerobic conditions, and they will accumulate lactic acid as an end product of metabolism, and that can lower the pH at the enamel surface and cause pitting of that enamel surface that can lead to caries or cavity formation. Right, and so here's just a shot of sort of the rate at which this plaque formation or biofilm formation can occur without brushing teeth. Right, we've all been camping or backpacking and forgotten our toothbrush, and you get this sort of fuzzy tooth feel. Right, the longer you stay out there without treating your teeth surface. However, the rate of dental plaque or biofilm formation is directly related to the diet, right? So if you have a higher carbohydrate diet, that plaque formation will occur much more quickly and can lead to more advanced or rapid rates of tooth decay without active brushing or removal of that plaque. However, if you have a higher protein, lower carbohydrate diet, those proteins result in slower growth rates, selection for organisms that are not growing fermentatively, and subsequently a lower rate of plaque formation and a lower rate of caries formation. And you'll notice that if you selectively choose to consume a protein-rich, carbohydrate-poor diet and don't brush your teeth for a few days, you'll notice that your teeth won't feel quite so fuzzy or as much sort of plaque formation versus sort of a high carbohydrate diet, low protein consumption for the same relative duration. Here we see a false colored transmission electron micrograph of a group of microorganisms that can be found in dental plaque, and what you see here is just basically a rich, diverse array of microorganisms as indicated by the different types of cell structures that are found in there, and also some tight associations amongst different members of that human microflora, as you see with the direct connection between some of those smaller rod shapes and larger rod shapes. It's very complex communities that can form in the human oral cavity. Here we see an electron micrograph of some of that biofilm formation that can occur with growth of cells on the tooth surface. You can see sort of the cells are pointed out there, and then that extracellular polysaccharide matrix that encases some of those cells, and that is the stuff that limits oxygen diffusion down to the cells and promotes a fermentative lifestyle. So moving on to another compartment of the human body, the gastrointestinal tract. The human GI tract, right, consists of a couple of different components: stomach, small intestine, and large intestine, responsible for digestion of food, absorption of nutrients, that sort of thing. It is very densely colonized by microorganisms, right? Those are huge numbers. That's 10 to the 13th or 10 to the 14th total number of cells in the lower and upper gastrointestinal tract. There are differential distribution of those organisms in the GI tract that we'll talk about now. And the reason there is differential distribution of organisms is oftentimes due to one major controlling factor, and that's pH. The secondary controlling factor would be redox. The stomach is highly acidic, right, and that limits the kinds of organisms that can make a living there. And we'll see their pH plays a major role in structuring the communities of other components of the gastrointestinal tract. There are functions or roles of these organisms. They're not just commensalistic organisms. They play a mutualistic role between the human host and proper functioning of the gastrointestinal tract. And this is that they carry out, as there are a suite of characteristic traits or processes that these microorganisms carry out, and much of this has to do with vitamin production. Things that are not commonly found in our diets are produced by members of the gastrointestinal flora. We've talked already about potential prevention of disease, as well as proper development of the immune system and circulatory system in the gastrointestinal tract can be directed by these organisms. And so this is in addition to those other traits we talked about already. So here's a shot of the human gastrointestinal tract and a list of the major bacteria that are present. And what you see is, as we move from the stomach to the small intestine to the large intestine, we sequentially increase the pH, where it becomes more and more neutral. And as we do so, there's a major shift in the distribution of the types of organisms that are found in these different compartments of the gastrointestinal tract. And you get an increase in species richness, a significant increase in species richness as you move from the stomach to the colon, transitioning from a pH of 2 through a pH of 4.5 in the small intestine to a pH of 7 in the large intestine. Here we see some of the roles of these members of the intestinal microorganisms, right? So some of them produce gas, Others produce some of the odors that are associated with that gas, and others produce enzymes that help us digest certain carbon substrates. Some of them also play a role in modifying steroids that our circulatory system will carry as part of our endocrine system, and these organisms can play a role in modifying the structure of those organic molecules and re-releasing them to the circulatory system so they can play subsequent roles in our endocrine developmental process. They also play a role, skipped ahead there. They also play a role in vitamin synthesis that we've mentioned already. So here's some shots of associations of intestinal bacteria, the bacterial cell wall, or the intestinal cell walls, and what you see in that middle slide, I'll use the pointer here. This is the epithelial cell lining, so these are villi coming off of epithelial cells, and these are individual bacterial cells associating directly with those villi or a little bit further away from that cell surface. And so you can see there's a sort of a high density of microorganisms associated with that intestinal cell wall surface, and the distribution of those organisms varies throughout the gastrointestinal tract we've talked about already, as well as the diversity of organisms that make those associations. So let's look at the distribution of some of these organisms in the upper and lower respiratory tract. We've talked about some disease-causing organisms already. Let's talk about some members of the normal flora in these compartments. There's only a restricted number of organisms that colonize the respiratory tract. We've talked about some of these already that are found in the upper respiratory tract, and those genera are listed there. Lower respiratory tract typically lacks microflora, except for disease-causing organisms in unhealthy individuals. If we look at the bottom end of the gastrointestinal tract, or like closely associated components of the human body that are part of the urogenital tract, you can see some characteristic structures there for both males and females. The bladder in both males and females is typically sterile unless there's a disease process occurring. So we've talked about some sexually transmitted disease that cause urethritis. Those would be organisms that might be found in an infected bladder but not in a healthy one. However, as health conditions are altered, you can see a change in the pH of the bladder that can make it a suitable site of colonization and growth of things like E. coli and Proteus mirabilis, which are frequent causes of UTIs in both males and females, and more commonly in women. That brings us to the end of our discussion of members of the human microbiome that are part of the normal flora. I'm going to stop the recording now, and there'll be a separate recording that talks a bit about some other components.